Okay, so I'm recording now, and I wanted to say that I will be sending out an email that gives some tips and strategies on how to approach studying for this next exam that's a week from today, and kind of give you some pointers of how to organize your study notes and the study slides and start grouping the information, I guess, for lack of a better. So let me give you an example. So in the mineral nutrition, let me share my screen. In the mineral nutrition slide set, I did say, please know this table 4.2 in the sense of understand how they group, the different minerals group into different categories of how they behave in the plant, what are their functions. And so grouping them in this way as you're studying will be important, because I will ask, so this is multiple choice ideas, okay? So think about this idea where I might pose a multiple choice question where I'd say, this nutrient is important in energy storage or structural integrity, and it can be part of the cell wall structure, and so what I would do is give you several bits of information about that nutrient, and then I give you a multiple choice, A, B, C, D, pick which one I'm talking about. And so between what you're reading on this table for what's going on with these nutrients, and then let me go back to the other, further into this slide set, we got into I covered several of the minerals in the slide set that I typed up as well. So then looking at table 4.2 and then looking at some of the information for the ones I covered anyways, you can combine that information. And so, what I'm saying here about potassium, these types of things would show up on the multiple choice. Like I'd say this mineral is highly soluble. It causes lodging of plants when you have deficiencies. Then I might include highly mobile is another idea. And then going back to table 4.2, I might include some of the information about potassium from that table too. So it'd be included in one, multiple choice question, and then I'd give you 4 minerals to pick from. So that, I think, is the best way to study for this. These questions on the minerals. And remember, I also asked you, I might give you a list and say, which of these are immobile? And I'd give you, of course, one would be immobile and the rest would be mobile. And then I might ask, which one on this list is a macronutrient? All right. Did someone have a question? Okay, so this idea of, I would give you several clues in the question. So I'm not asking you to memorize all the content for every element, but if you are learning some of the key things, I'm sure in the question you'll be able to pick, oh yeah, I remember studying that. I remember that one. And so kind of consolidating table 4.2 and then going down further in the slide set with my write-up of these different minerals. And some of the unique things about some of these minerals, like iron, we said technically they classify that as a micronutrient, but it's the one that's required in largest amounts. So again, that's something very unique about iron. So that would be an easy thing. If you saw that in a question, you're like, I know that one. That's the one that's iron. And so these types of ideas, look for unique things about the minerals, so that only that mineral, that's the one she must be talking about in her description. So that would be a good thing to start organizing in your head in that regard. So I will send out an email. I'll say this again in the email, but then I'll give you some other tips on how to strategize you know, what would be more appropriate for essay style question, because usually it's the essay questions, of course, that are higher point value per question compared to multiple choice. So, okay, so I will send that out this afternoon. And I already sent out one email just saying this is the content for the exam. So hopefully you saw that in your inbox by now. Okay, where? Did someone have a question at all? I thought I heard something. Let me go to, I guess this one. I'm going to share. I don't want that. Let me stop share for a second. It's doing it again. Let me switch it. Share screen. Come on. All right. Let me get this set up. So hopefully you're seeing one big slide. Is that correct? Of the light reactions?
Yes, ma'am.
Thank you. All right, so let's review a little bit of what we covered so far with the light reactions. We are, typically you start with photosystem 2, so when you're studying it, think about this process from the kickoff of photosystem 2. And we have the light energy striking the antenna pigments around the photosystem, and those antenna pigments are absorbing that light energy, and then through inductive resonance transferring that energy from pigment to pigment until it finally funnels into the reaction center, where you have this reaction center chlorophyll that will absorb that light energy, and then one of its electrons gets boosted to an excited state. And if everything's going okay and it stays in the triplet state long enough for field fighting to grab it, then that's where you have the true example of, now we can say light energy has been converted to chemical energy, because we have a redox reaction happening where an electron finally gets donated off from that chlorophyll in the reaction center, in the P680 reaction center, off to field fighting. So then remember, now this, it's kind of like a game, because now chlorophyll is like, I'm missing an electron. I need an electron to be chemically stable. I need one. Who can give me one? So water steps up to the plate, and through the oxygen evolving complex, that's part of photosystem 2, and the absorption of light energy, part of that absorption of light energy also allows for the oxygen evolving complex to rip electrons off of water, which is very hard to do. And so the electrons get stripped off, water gets oxidized, and that electron from water replaces the lost electron that the chlorophyll is trying to, solve that situation. So the chlorophyll will turn to water and say, give me one of your electrons so I can fulfill and have my full complement of electrons. And then you have to imagine this is happening in like picosecond timing, where now chlorophyll can get hit with light again, and its electron gets excited and boosted off to field fighting. Okay, so this keeps happening very, very quickly. When you look at the electron flow process, we said that field fighting, let me go to oxygen evolving complex. Where is, all right, let's look here. So the field fighting moves off of photosystem 2, starts interacting, well, actually it doesn't move very far, because plastohydroquinone is usually right there, and field fighting will transfer the electron to the first plastohydroquinone, and then that one quickly turns and says, I'm going to give it to the second plastoquinone. And so that happens, and then it has to all repeat all the way back to photosystem 2 getting hit with light energy, the electron from the reaction center chlorophyll excited, given to phenolphthalein, then water gets stripped of electrons, electrons given to the chlorophyll, phenolphthalein moves to plastoquinone, does this all over again. So if you repeat all of that, then that second plastohydroquinone will end up with 2 electrons. And that's good, but it's not quite finished yet, because in order to be chemically stable, that plastohydroquinone can't just pick up 2 electrons. It has to pick up 2 protons as well, and it'll grab it from the stroma side. So where it grabs it from, it is important. You need to know that, because that is playing a role in the development of a proton gradient. So the plastohydroquinone grabs the 2 protons from the stroma and says, okay, now I'm happy, I'm fully reduced, I'm stable, I'm now plastohydroquinone in the reduced state, and can do my next job in this whole game of the electron transport chain process. So we looked at this, that you need this 2 electron gate, you need 2 electrons and then 2 protons to get a fully reduced plastohydroquinone. All right, so plastohydroquinone, as you can see, it's actually the top structure here. They just zoomed into the benzene molecule here, or the ring structure here. So here we see it is still a fairly small molecule, and it is hydrophobic, so it can stay in the hydrophobic portion of the membrane, and so it releases away from photosystem 2 and where phenolphthalein is located, and starts to move laterally in the membrane until it bumps into cytochrome B6F, another big complex that cytochrome B6F really can't move. So it's up to these mobile carriers to do the job of shuttling electrons. So PQH2 comes along and says, here, I'm going to give you 2 electrons, but I'm going to put them into different paths. So one goes linear and will end up at photosystem 1, and the other one gets given to the cytochrome B in the cytochrome B6F and kicks into the Q cycle. And so we talked about that as well. And here we see the first PQH2 is coming along and says, okay, here's 2 electrons, one goes linear and the other one goes into the Q cycle, where cytochrome B gets it, and then it passes it quickly to another cytochrome B, who then quickly gives it to a plastoquinone. Well, now plastoquinone has 1 electron, so it's semi-reduced. That's not good enough. Plastoquinones are greedy for electrons. They said, sorry, I need another one. And so that necessitates that you have to go all the way back to the beginning again and have photosystem 2 hit with light energy, and all that happens again so that you can get another plastohydroquinone coming along and bumping into that same cytochrome B6F and donate off 2 more electrons. So then, once that happens, that semi-quinone will get a second electron and says, great, now I just need 2 protons, and I'm going to grab them from the stroma again, and then become PQH2, fully reduced and in its stable form. And then it releases from the B6F complex, and they don't quite know what happens to it after that. Presumably, some people think that it dumps its protons over into the LUMA right away. It's really hard to track, and so they're still working that out. So right now, we're just going to say it releases from the B6F and just kind of stop there. That's fine. But when you look at the Q cycle and you see what it's doing, and then if you imagine like getting rid of it and not having the Q cycle as part of the process, you see that, oh, there's proton pumping going on here, that if we got rid of the Q cycle, those extra protons probably would never get pumped across the membrane. So really it seems that this evolved to ramp up a proton gradient to allow for protons to get pumped across the membrane during this process. And again, anytime you see a proton gradient establishing across a membrane, that should ring a bell in your head. I bet this is going to have something to do with ATP synthesis. So I think we left off right around here last time, thinking about this idea, and the Q cycle. So let me just pause for a second. That was what we just talked about, was all review from last time. Are there any questions about any of that content before we move into the linear path?
Can you hear me?
Madeline, yes, I can hear you.
I'm confused about what's going on.
Oh, now you're breaking up again.
No, I'll put it in the chat and then.
All right. Yes, please chat. Is there somebody else who wants to
So,
ask?
does it only take one photon to excite photosystem 2? How many photons? Do you know any about that?
I don't know. I think I got a fruit fly flying around me again. I think, I don't know if your book gets into that. I think somewhere in there they talk about the quantum energy that's necessary to be absorbed, but I don't have that memorized. It's not something I routinely pay attention.
Yeah, it was worth a shot.
Yeah. So check the book. See, I thought somewhere in there they talked about the quantum energy being absorbed, and they have figured out, they've done some of the thermodynamics and the math and all that, but I have not kept track of that. Other questions? Okay, let me just check the chat. Okay, so just asking for clarification on the Q cycle and the what's going on in the cytochrome B6F. So let me go to this slide. So this is showing both when the first PQH2 comes along on the top and when the second one. So let's start on the top. So the PQH2 that's fully reduced moves away from photosystem 2 and phenylphytin and moves laterally and bumps into cytochrome B6F and donates 2 electrons. One goes linear, and then the other one goes into the Q cycle. So the Q cycle, if I say, what is the Q cycle? You would start with, okay, the plastohydroquinone has bumped into cytochrome B6F. One of the electrons gets given to cytochrome B, which gives it to another cytochrome B, which then gives it to a plastoquinone. Now we have plastoquinone in its semi-reduced state. It has 1 electron. We know plastoquinone is greedy for electrons. That's not enough. We need 1 more. So you have to imagine all of this happening all over again, all the way back to photosystem 2 getting hit with light energy, so that we can get another plastohydroquinone coming along to that same cytochrome B6F, and again donating 2 electrons. 1 goes linear and 1 enters into the Q cycle, which means it goes to the first cytochrome B, the second cytochrome B, and then that semi-reduced quinone gets its second electron. So that's good. It's like, yay, I got 2 electrons. Now all I need is 2 protons to be fully chemically satisfied. And so you see the protons being grabbed from the stroma. And then plastohydroquinone is fully reduced and gets released off the B6F complex. So that's the Q cycle.
Thank you.
Yep, you're welcome.
Is there any other complexes that are pumping protons across the gradient, or is this the one?
So we have proton pumping here, and then the other way that protons build up through this process is when water is getting stripped of electrons. We have leftover protons, but it's not when we have leftover protons from water, those aren't getting pumped. They're just building up in the lumen. So we'll get into that. I'll show that part of it too. And so this Q cycle is really where you see the idea of pumping across the membrane of the protons. Okay, so let's think about the linear path now. So these PQH2s that have come along to the cytochrome B6F, they give an electron out to the linear path. And so who are the players? Well, that iron sulfur gets it first. And that's, we're still in the cytochrome B6F for that. So the iron sulfur grabs it, quickly gives it to cytochrome F. And then cytochrome F says, I hope there's a mobile carrier here. I hope plastocyanin is here, because that's the next step. So plastocyanin is, kind of hugged up to the cytochrome B6F complex, gets that electron, and then its job is to be a mobile carrier. And the way they've drawn it here, again, it's associated with the thylakoid membrane, but on the lumen side, and kind of moves along that portion of the membrane until it can reach photosystem 1, which again, another huge complex. It's a transmembrane complex, way too big to be super mobile. And so plastocyanin has a very important role because it's little tiny zippy protein that can zip along. I think I told you I imagine those roller coaster rides where you're hanging below the track, swinging and zipping along. I can't imagine. Anyone who's done that, I give you credit. I'm too chicken to do that. So here we are, plastocyanin comes along, bumps into photosystem 1. And now we're at a critical juncture because photosystem 1 can take that electron from plastocyanin, but only if it's already been hit with light and one of its electrons has been in the chlorophyll reaction center, one of the chlorophyll pigments has lost an electron. Otherwise, plastocyanin has got to sit there and just kind of hang out and wait for all that to happen. So this is, I'm trying to get at the idea there that we have to have a good, smooth regulation of the light reaction so that both photosystems are getting hit with the necessary light energy to keep them working in concert, so that by the time plastocyanin bumps into photosystem 1 is like, yes, give me that electron, and is able to just keep things ticking along. So I believe we left off here looking at this process where in the linear path, the plastohydroquinone gives an electron to iron sulfur and cytochrome F. Okay, and that's all within the cytochrome B6F complex. And then the cytochrome F gives it to the mobile carrier plastocyanin, which moves along, hopefully finds photosystem 1. So here it is again, where plastocyanin's moving over to photosystem 1. Well, in the meantime, photosystem 1, we sure hope, is getting hit with light energy. Antenna pigments absorbing that, transferring that energy via inductive resonance, funneling it, so to speak, to the reaction center chlorophyll. That chlorophyll pigment absorbs all that lovely energy. The electron gets excited, gets boosted to the acceptor, which we'll hold there for a second. And then now chlorophyll in the reaction center going, okay, I'm missing an electron. I really need one. And that's when plastocyanin can step up and say, I have one. I will give you the electron to fill the missing, the gap there. And so if all of that happens, then plastocyanin can do its job of donating an electron to that chlorophyll in photosystem 1's reaction center. Okay, so if photosystem 1 is operating correctly and that absorption of light energy excites that electron in that chlorophyll in the reaction center, who gets it? Let's look at that. So when light energy is being absorbed by the reaction center chlorophyll, this is how you would denote it on the exam. So if you say P700 asterisk, you're telling me you understand that chlorophyll in the reaction center has absorbed light energy and now it's in an excited state. That electron has been boosted and is in an excited state. And if everything is going okay, then that excited electron can be donated off to an acceptor molecule. And then you would write it this way, where if you write it as P700 plus, you're telling me I know that chlorophyll donated off its electron. So now it has a positive charge, which means it's ready to accept an electron from plastocyanin. All right, so these electrons need to keep going, though. We still have to finish the thought here of how do they finally end up to the ultimate and final electron acceptor. So when you peer inside photosystem 1, you find that there's many players in the game here that will take that electron and pass it on down the line from donor to acceptor, donor to acceptor. So we need to kind of zoom into photosystem 1 and say, who's playing the game in that portion? So let's look at that. The first acceptor, so when that chlorophyll in the reaction center, when its electron gets all excited, it's going to give it off to another chlorophyll, another special chlorophyll molecule called A0. Then that A0, and you can just write it as A0. A1 will get it next. That's a special phyloquinone molecule. It's actually vitamin K if you look at it, which then gives it to an iron sulfur protein, and you can denote it FES. That's fine. And then there's another, actually you should denote it FESX, sorry. So the first iron sulfur protein that gets it, denote that it's an X, because the next one that gets it is an A version, and then another one is a B. So you have the electron going to A0, then to A1, then to FES-X, FES-A, FES-B, all in super rapid succession. We're talking picosecond transfers here, and you get these, you know, the electrons going from donor to acceptor, all the way down. So let's look at this visually, where they put it into context of this whole photosystem 1 reaction center. So here's the P700 is the special, you know, it's the reaction center where there is that special chlorophyll, and its electron got excited and boosted and given off to A0, then to A1, then to FES-X, FES-A, FES-B, and then finally it's going to show that it's going to get given to this ferredoxin molecule. And you can see ferredoxin is just hanging out next to photosystem 1. It's not actually an integral part of the photosystem. It's just bound to it, waiting for its turn to get an electron. Okay, so there's quite a little bit of transfers going on here within photosystem 1, and I do need you to know all the players and the order. Okay, so once you have that process, then ferredoxin will be sitting there going, okay, I'm ready to take one of those electrons coming through here. And ferredoxin is a protein. You can abbreviate it FD. Ferredoxin is quite a versatile protein, and it's quite good at giving off electrons to appropriate reactions. And so when you look at it too, it's important that you recognize yeah, they do show it here. Ferredoxin is found in the stroma. I do need you to know that, because that will help you understand that it can do several things in the stroma. So there's several different things happening in the stroma where ferredoxin can play a role. But let's just focus on the light reactions. What is it doing in the light reactions? So it grabs an electron from FES-B, and it's small. It's a small protein. It's only lightly associated with the photosystem. So once it grabs an electron, it can release. It can release off of that, and its job, and that's what they're trying to show here, that the ferredoxin is released. And then it has to do its job of getting that electron to where it's needed next for the light reaction. So what is that? It has to find the FNR protein. And it used to be called a flavoprotein. FNR used to be called flavoprotein, but on the newer editions, they've renamed it. And so now, actually, they named it to something that is nice. It actually makes sense. They named it ferredoxin NADP reductase. So the FNR protein, the ferredoxin part of the name means that it associates with ferredoxin, and it's going to grab an electron from ferredoxin, and then the NADP reductase part of its title means that its goal is to give that electron to NADP. It's going to reduce NADP. So it's a reductase protein or enzyme. Grab an electron from ferredoxin, give it to NADP+. So that's why they named it ferredoxin NADP reductase. So that's the job of that very special enzyme. It's going to then find NADP+ in the stroma and say, hey, you're the ultimate final electron acceptor in this whole process. Here you go. Here are some electrons. And then once NADP+ accepts that, you can denote that it's been reduced by writing NADPH. Right? So now it's been reduced. So NADP+ is the final electron acceptor. And make sure, if I ask that question on the third exam, make sure you add the plus, because, and it's, you know, if I say, what's the final electron acceptor? It's NADP+. If you write NADPH, that's wrong, because it's already reduced. So make sure you understand what form is the final electron acceptor in when, you know, when it's about to take an electron. NADP+. So let's look at that. So here, if you are curious, this is NADH. The phosphate would just go on, let's see. Oh, they're just showing part of it. So here is NAD Okay, so here on the right is NAD+, nicotine amide adenine dinucleotide. So that's why we abbreviate it to NAD. So here's the whole structure. And then if you want to make it NADP, you just add the phosphate right here. And so this whole molecule is waiting at the very end of the light reactions. And when it gets its electrons and grabs protons, it will convert to this. They've just abbreviated it over here now. They're just focusing in on where you see the protons and electrons getting added to this portion of NADP. All right. So if you're kind of have a chemistry brain and this helps you see what's going on, I added that in. Okay. So that's happening in the stroma. So here we have photosystem 1, and they don't show all the players in this little drawing, but make sure you understand there's a bunch of players in here. And then finally ferredoxin gets the electron, releases from photosystem 1, finds FNR, gives the electron to FNR, and FNR says, hey, NADP+, I got this electron. And so then you have the final transfer to NADP+, and you get lovely NADPH, all happening in the stroma there. And that's important that you understand that's where NADPH is getting made. Questions on that portion of the linear path all the way through to NADP+? Okay, I guess not. Clear as mud. Hopefully it's clear, but all right. So you start looking at this and you realize there's some other stuff going on because we have this huge, wonderful ATP synthase protein complex. So we need to think about that. Well, you see protons are funneling through that ATP synthase. So we need to go back to the idea of proton pumping. We said with the Q cycle, we have protons disappearing from the stroma and getting dumped in the lumen. So that is where we have significant proton pumping going on. And then the other way we get more protons showing up in the lumen is when water is getting stripped of electrons, because if you take electrons away from hydrogen, you're left with protons. So now you see this buildup of protons in the lumen, and you now have this proton gradient that's getting established. And so then immediately think about, okay, this is potential energy now that can be used to do something. When we speak about the light reactions, it's the idea here is that that proton gradient will be used so that those protons will want, chemically speaking, through diffusion processes, will want to diffuse across the membrane to equal out the concentration, but they have to go through the ATP synthase to accomplish that. And when you have them flowing through this amazing synthase complex, it actually creates a torque force, a rotational torque force, that causes the ADP and the inorganic phosphate to come into contact with each other and react, and you get the synthesis of ATP. And so they describe ATP synthase as a little molecular motor, and that's very, very true. It's just amazing to think this is happening in all of our bodies right now. Well, we can't do photosynthesis, but we have ATP synthase. So this is cooking along in your cells right now if you ate a decent lunch or breakfast, I hope. All right, so what I just described, they have to give it a jargon for us to memorize, of course. So they call it linear photophosphorylation. The linear just means that you have a linear electron transport chain process that is tied to the establishment of a proton gradient. And then it is all happening because of the absorption of light. So that's where the photo comes in. So they're, specifically speaking to, there must have been some kind of photosynthesis going on for this to happen. And then phosphorylation, that term, is just referring to the ADP and the phosphate reacting with each other. ADP is getting phosphorylated to make ATP. So linear photophosphorylation, when you hear that term, what they're describing in just that two-word statement is the idea of this proton gradient that gets established through the light reactions of the electron transport chain. And as those protons diffuse through the ATP synthase, that allows for ADP to get phosphorylated, and you get the synthesis of ATP. So I remember when I first started learning the light reactions, especially to the level of detail we just went through, and it's complicated. It's like, wow, there's a lot of steps to this. Why didn't they just, through evolutionary processes, simplify that a little bit? But when you start looking at the thermodynamics of this whole situation, it really is quite a beautiful process to allow for free energy to develop in a way that's controllable by the plant. If you look somewhere, I read in actually a couple different reputable sources, that if you look at the whole thermodynamics of this and you shorten up the light reactions, the amount of energy that would take place in one fell swoop, and if you shorten the reactions up, it would probably cause the cell to, you know, burst into flames, and it would be so much energy release all at once that it would just be the death of the plant. And so you have to think about, you know, there needs to be a way to control this process so I can have some free energy to make ATP, but I have to do it in a way that doesn't allow me to burst into flames. So what we see here is that as the electrons are getting transferred from donor to acceptor, on down the line, we say it's a downhill process. What we mean by that is that it's energetically downhill. It's a negative free energy process as you work your way along the electron transport chain. What that means is there's a little bit of this free energy now that can be used to do something else in relationship to this electron transport chain. And that something else is the proton pumping, to get the protons pumped from stroma to lumen and get your gradient, your proton gradient established. So that free energy is allowing for the proton gradient to get established. And so that's why I tend to say, you know, when you see an electron transport chain process happening in a membrane system, chances are you're linking that to a proton gradient. And that's not unique to the light reactions of photosynthesis. That's also happening in our mitochondria and in the plant's mitochondria. They do this too. And so if you see electron transport chain processes happening in a membrane, proton pumping, you look for that right away. You're like, I bet there's proton pumping going on, because some of that free energy, as those electrons are playing the game of moving from donor to acceptor, part of the goal, the way you win the game, is to get those protons pumped so you can get that lovely gradient, so that allows for ATP synthesis. So you have proton pumping through this process of electron transfer, but don't forget, you also have water being split, which is also increasing the proton amount in the lumen. So you actually have protons coming from two different mechanisms, so to speak. Okay, so that's how you think about this idea of, yes, the light reactions, you certainly have this electron transport chain process, and it has a goal of getting NADPH formed, and that's great. So that's one way to win the game, but that's not the end of the story. That whole process is also setting up the situation quite nicely for proton pumping, so that you can get a proton gradient, which lets you have ATP synthesis in this process as well. Questions on that concept?
I have
JZ, I can't hear you.
I have a quick one, but it kind of relates to microbial ecology. Would for example, the photo, would it be a cyclical photo phosphorylation in the purple bacteria, for instance?
Yes, you can think of it that way, because yeah, with photosystem 2, when they only have photosystem 2 and they're doing that circular flow, then technically you'd say, yeah, that's circular photophosphorylation in the purple bacteria. Yeah, which hold that thought of circular or cyclical, or I'm not quite sure which word is better. We're going to get to that in a second with regard, because sometimes plants can do this funky little thing that I'll talk about. Here's the image that they've worked at very hard to dissect the ATP synthase complex, a multi-subunit complex, and just an amazing, beautiful structure where you have that proton flow through, and then they're trying to show here with this little winging arrow here that you have this rotational torque force that causes the kind of, not like true spinning up here, but it allows for these subunits to interact with each other in such a way that you get the ADP in the phosphate reacting to get you ATP. And then this I put in here just to remind you that ATP is great, and we talk a lot about ATP, and certainly it's appropriate. Don't forget though that there are other molecules that have a decent amount of free energy available if you cleave some of their bonds, for example. And so ATP here, you know, the reason why it's such a good energy molecule is that whenever you see those squiggly little bonds, that means it's a high energy bond. So when you cleave that, you can make good use of the ATP molecule to drive other reactions. And so here ATP in this chart, this table, you see ATP has a free energy of negative, let's just round up, negative 32. That number is a magic number I want you to memorize. All right, negative 32. That will, it's a good one to know. All right, so ATP has a free energy amount of negative 32 kilojoules per mole. But you can see that there are other molecules that are actually higher. They have better free energy. It's just, so keep that in mind. There are other molecules out there. ATP is heavily used by all living organisms, but it's not super long-lived. And so it you know, like when you think about the light reactions, they constantly need to make ATP because it's not like we have a storage of ATP in the plant body or in our bodies. We don't have anywhere to store ATP. All right, so it's constantly made, constantly used. You have this whole process going on. So just keep that in mind. Okay, this is actually taken from one of my microbiology textbooks. That's why they're, but it happens in plants and higher organisms too. Okay, let's get to this idea of cyclic or cyclic circular photophosphorylation. Plants can also do this. So what do I mean by this? In your book, does touch on it, but they don't really give a picture. So I drew this to try to get you visually thinking about it as well. What does that mean? Well, what they're trying to tell you is that the electrons go in a circular path. So let's go back to this picture. You've seen this already. So we know, even though I talk about the light reactions all neat and tidy, where everything's really close to each other and they're right next to each other and that's how it goes. Remember, that's not reality. You have clustering of where you find photosystem 2, where you find photosystem 1 in the thylakoid. So now you have to imagine parts of the thylakoid membrane can break off, not really break off physically, but they can start doing their own thing during certain, certain sections of the thylakoid. What they can do is sometimes the plant will say, you know what, I want to kick in, I want to do some cyclic photophosphorylation. There's a situation arising that I'm presented with due to my environment and what's happening to me. I'm gonna vote for doing some cyclic photophosphorylation in this section of my thylakoid, but I'll keep clicking along with the light reactions, the linear photophosphorylation in other parts of the thylakoid. So the plants have this flexibility. What is this? So there's electrons. So this is plastohydroquinone coming along to cytochrome B6F. So this oval that I drew is cytochrome B6F. And the plastohydroquinone gives its electron into the linear path, like we've already discussed, over to photosystem 1. Now what happens is photosystem 1, instead of giving the electron off to ferredoxin, it backs it, or I'm sorry, after it gives it to ferredoxin says, you know what, I'm not going to give the electron to FNR. I'm going to take it back to cytochrome B6F and the plastoquinone there. And so it starts this circular flow where ferredoxin decides, so to speak, you know what, in this little section of the thylakoid, I'm going to circle back and give the electron back to the plastoquinones and the cytochrome B6F and just keep zinging around the electron round and around and around. And you look at this and the way I drew it, it's like, okay, so why would they do that? What's the payoff? There needs to be a payoff for this to evolve. So what is the payoff? Well, if you look at, let's put the circular flow here. So here's ferredoxin. So imagine ferredoxin saying, nope, I'm going to go back over over here, the cytochrome B6F and PQH2. And I'm just going to do this section over and over and over, keep zinging around. Well, if you look at what's happening in this section of the light reactions, there's something very important happening. And what else is happening here?
Proton pumping.
Yep, proton pumping. So these ingenious little plants, through natural selection, of course, and adaptation, they said, you know what? I need some extra protons because I really need ATP. I've noticed my ATP levels are dwindling. Remember, you can't stock up on ATP. So the plant's like, I'm in a situation where, oh my gosh, I don't have enough ATP. I'm going to kick into circular flow here so that I can really ramp up proton pumping, and that should get me more ATP really quickly, which can be done temporarily. So it's not like they're always doing this. They do it when their current environmental situation and physiological situation is kind of demanding, I need more ATP. And so they'll kick into this. And so go ahead.
That thing that's happening like lower in the canopy where the leaves aren't getting as much full sunlight?
Yes, it can happen under those conditions. Yes, and they have measured that. And that's one good reason why a plant would want to do it, right? If I'm getting shaded out and I'm just, I'm not getting much ATP anyways, I'm going to kick into the circular flow to kind of ramp it up. And so when you look at that little trick of ferredoxin allowing for the circular flow in certain portions of the thylakoid, it's to ramp up proton gradient, which will ramp up how much ATP you make. And the downside is you don't get any NADPH made, but don't forget the rest of the thylakoid membrane is probably very busy still doing, light reactions in the normal way. So it's not a huge deal here. The payoff is huge for the idea of I can make extra ATP very quickly because I'm ramping up my proton gradient. And also there's no water split. They don't, in this section, they don't see it. So it's really focusing in on, I just want to pump protons so I can get more ATP. And so another reason a plant might want to do this is if they are, well, I guess we'll get into that. When we get into carbon fixation, we will learn that some plants aren't as efficient at carbon fixation than others, and so they need extra ATP. Well, I shouldn't say they aren't as efficient. I shouldn't really say it that way. They just have to do the carbon fixation a little differently because of where they adapted to live. And so they do have a little higher need of ATP. And so those types of plants will tend to run cyclic photophosphorylation pretty frequently in order to deal with their extra ATP needs to fix their carbon. And let's just finish the thought here on this one last slide. You worked with DCMU in the lab. I understand that yesterday's lab, the light reactions did work. Is that true? For the most part anyways, you were getting some good data. Does anyone from Group B want to comment? No? I heard it all worked. So
I think Group A was in lab yesterday.
Oh, Group A. Okay, whichever group. I'm sorry. So I heard it worked. Is that true?
For the most part, yeah, we got some really good data.
Super. Yeah, I was thrilled when I got a text. It's like, yes. So I'm glad at least half the class was able to see. So DCMU is one of the treatments you worked with, and this is really what you were doing. When you added DCMU to those chloroplast suspension, you were messing with the light reactions in a very particular way. You were messing with the plastohydroquinone, being able to deliver its electrons. And so you had a very potent way of messing up the light reactions. And of course, the higher the dose of DCMU, the worse it got. Did I hear a question?
That was the pesticide or the herbicide?
I didn't hear everything that you said, Madeline. You broke up.
That was the herbicide DCMU?
Yes, DCMU is the herbicide. Yes, the herbicide. Okay, it looks like we ran out of time. There's just a couple more slides in this slide set that I did want to address with regard to what happens if things go wrong during the light reactions and how does a plant deal with that. So besides being hit with DCMU, there's some other things that can happen that can affect the light reaction. So we'll talk about that next week. So look for an email from me this afternoon talking about exam 2, which will be given next Friday starting at noon. And as I make the exam, I can give you some more feedback back as well, but I'll at least send out an email today giving you tips and strategies for how to study for that, okay? So stay warm and dry, everybody, and try to have a good weekend.
You
So,
too. Thank you.
Thank you.
Have a good weekend.